Good morning, ma'am. My name is Aldrin Dave Diacinto of 3 A1-8, and this is my midterm laboratory exercise DNA analysis, and the lesson objectives are: explain the importance of DNA analysis, enumerate the step in DNA analysis, and on my presentation, I will discuss also the DNA, its location, composition, and origin, forensic DNA analysis and how it used to solve a crime, and what are the steps in forensic DNA analysis, and last, what are the types of DNA evidence analysis. What is DNA? The DNA is molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instruction for the development, functioning, growth, and reproduction of all known organisms and many viruses. Researchers refer to the DNA found in the cell's nucleus as nuclear DNA. An organism's complete set of nuclear DNA is called its genome. Besides the DNA located in the nucleus, humans and other complex organisms also have a small amount of DNA in cell structures known as mitochondria. All nuclear organisms have double-stranded DNA genomes. We are reasonably sure now that DNA and DNA replication mechanism appeared late in early life history, and that DNA originated from RNA is an RNA protein word. What is the forensic DNA analysis? DNA profiling is the determination of a DNA profile for legal and investigative purposes. DNA analysis methods have changed numerous times over the years as technologies improve and allows for more information to be determined with the less starting material. DNA analysis is the most powerful tool for human identification and has clear forensic application in identified testing, crime scene, and mass disaster investigation, and percentage determination. The development of forensic DNA technology in other countries and its potential to improve the Philippine criminal justice system are briefly discussed. The utility of forensic DNA testing in criminal investigation was highlighted using an actual criminal case wherein DNA evidence plays a clear role in the resolution in the case. The steps in DNA analysis. The isolation of DNA from an evidence sample containing DNA of unknown origin, and generally at a later time. The isolation of DNA from a sample or e.g. blood from a known individual. Second, the processing of the DNA so that test result may be obtained. Third, determination of the variation in the DNA test results or types from specific regions of the DNA. And last, the comparison and interpretation of the test result from the unknown and known samples to determine whether the known individual is not the source of the DNA or is included as a possible source of DNA. And the steps involve collection, specimen storage, extraction, quantitation, multiple PCR, STR typing, interpretation of results, database, and calculation of match probability. The types of DNA evidence analysis has four types. First, polymerase chain reaction or PCR. Second, short tandem repeat or STR. Third, Y chromosome. Fourth, mitochondrial DNA. What is polymerase chain reaction or PCR? Is a method widely used to rapidly more millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to the large enough amount of the study in detail. While The short tandem repeat or STR is a common molecular biology method used to compare allele repeats of a scientific loci in DNA between two or more samples. This method differs from the restriction fragments length polymorphism analysis or RFLP. STR analysis does not the DNA with restriction enzymes. Y chromosome. Y chromosome, the male specific part of the human Y chromosome, is widely used in forensic DNA analysis, particularly in cases where standard automosal DNA profiling is not informative. A Y chromosal gene fragment is applied for inferring the biological sex of crime scene trace donor. And last, the mitochondrial DNA is the DNA located in mitochondria, cellular organelles within eukaryotic cells that convert chemical energy from food into a form that cells can use, adenosine triphosphate.